Good afternoon. Thank you for tuning into my presentation, and thank you to the organizers for allowing me to participate in the CIS annual meeting. My name is Emilia Falcone, and I'm a clinician scientist at the Montreal Clinical Research Institute, or IRCM. And today, I'm going to be talking to you about intestinal inflammation in chronic granulomatous disease, where the microbiome and reactive oxygen species interact at the mucosal barrier. I have nothing to disclose. So the objectives of today's talk are to discuss the variability in susceptibility to colitis in mice with NADPH oxidase 2 or NOX2 complex defects, or CGD, to examine the role of the microbiota in mediating colitis susceptibility in mice with NOX2 defects, to elucidate the impact of NOX2 defects on the function of the microbiota, and to evaluate intestinal microbiome signatures in patients with CGD at baseline and after specific interventions. So, chronic granulomatous disease, or CGD, is due to defects in any one of the subunits forming the NADPH oxidase complex 2 or NOX2. The most common form of CGD is due to defects in GP91 FOX, which are inherited in an X-linked fashion and affect 65% of patients with CGD. The second most common form of CGD is due to defects in P47 FOX, which are inherited in an autosomal recessive fashion and affect 25% of patients with CGD. Patients with CGD in general suffer from recurrent infections and inflammatory dysregulation. In fact, almost 50% of patients with CGD have some sort of gastrointestinal inflammation. When we evaluated the cohort of patients with CGD followed at the NIH, that's exactly what we found. 44% had gastrointestinal complications. However, it is interesting that this would mean that 50% of patients with CGD do not have gastrointestinal inflammation, leading us to wonder about other extrinsic factors which could be impacting susceptibility to gastrointestinal inflammation in patients with CGD. When we talk about gastrointestinal inflammation in CGD, for the most part, we're referring to a form of inflammatory bowel disease that shares common features with Crohn's disease, as well as with ulcerative colitis. From here on, we'll refer to this as CGD-associated inflammatory bowel disease, or CGD-IBD. The management of CGD-IBD is quite similar to the management of conventional IBD. That is, patients are treated with steroids, mesalamine, then followed with the addition of azathioprine. And if the IBD is still refractory, we proceed to more intense immune modulators, biologics, which do come with some risk in the context of an existing immune deficiency. And when IBD is still refractory, we're left with surgery or proceeding to hematopoietic cell transplantation, which we know is curative. That being said, if you've paid attention to the title of the talk, you know that we're gearing towards the microbiome. And why would the microbiome be important in this context? Well, it is possible that we can harness the intestinal microbiome either through an intervention like an elemental diet or fecal microbiota transplantation, for instance, to either prime the immune system to a better response to immune modulators, or perhaps better prepare patients for the transition and the preparation to hematopoietic cell transplantation. So, intestinal inflammation in the context of CGD. There is certainly a paradox here in the context of NOX2 defects. On the one hand, one would think that high levels of reactive oxygen species are generally associated with more intense inflammation and even some forms of conventional inflammatory bowel disease. However, on the other spectrum, we have patients with CGD-IBD, who for all intensive purpose have a real reduction in phagocyte-derived reactive oxygen species formation. We don't know what the precise impact of this defect, this NOX2 defect in phagocytes really is at the level of the intestinal barrier. Could it be leading to an imprinting or a modification of the reactive oxygen species that are formed in the intestinal epithelium? we know that there are NOX2 homologs in the intestinal epithelium, such as NOX1, NOX4, and the duoxes. We also know that the microbiota forms its own reactive oxygen species, especially hydrogen peroxide. So taking this all together, we have yet to uncover how NOX2 defect really impact the overall ROS balance at the intestinal barrier. And we know that reactive oxygen species are essential to maintain the integrity of the intestinal barrier. So we aim to have a sort of ROS sweet spot. Now, when we think of this inflammatory dysregulation in the context of NOX2 defects, in the context of low ROS levels, presumably, we know that there are other contributing factors, at least based on studies that were done in peripheral blood cells. Certainly, there are skewed Th17 polarization of CD4 cells, increased production of IL-1 alpha and IL-1 beta by monocytes. We know that there's also defective autophagy and increased NLRP3 inflammasome activation. We don't have a precise notion of how all these mechanisms come into play precisely at the mucosal barrier. It's an area, though, of active investigation. But it is almost certain that in some form, the intestinal microbiota contributes to the propagation of some of these aberrant mechanisms that promote inflammatory dysregulation in NOX2 defects. So we'll proceed with part one of this presentation, which involves our work in mice. So we know that P47 and GP91 knockout mice do not spontaneously develop colitis. And we followed our mice up until they were 16 months old. And you can see here when looking at these histology slides that the colon looks really normal and comparable to that of a young wild-type mouse. On the other hand, studies of induced colitis in the context of mice with CGD have been conflicting. Some studies in GP91 knockout mice have showed that they do not have increased susceptibility to at least DSS colitis, while another has shown that there is increased susceptibility to TNBS colitis, another chemical colitis model. Similarly, in P47 knockout mice, one study showed that there is not increased susceptibility to DSS colitis, while another one showed that there is increased susceptibility to DSS colitis. A nice study in P47 knockout mice showed that these mice truly do have increased susceptibility to chemical, innate, and immune-mediated colitis, while P47 knockout mice seem to be protected from enteric infections. From our end, this is what we found. When we looked at P47 knockout mice who were homozygously bred, that is, they are maintained with their microbiome that is intrinsic to their genotype. Here we see in the DSS colitis model that P47 knockout mice have increased weight loss and they have worse histology scores. We also noted increased mortality and worse disease activity indexes. When we tried to rescue this phenotype using bone marrow transplantation, we found that replacing the hematopoietic compartment and the P47 knockout mice with a wild-type bone marrow really did not reverse this phenotype. As you see here, similar to the P47 knockout mice, the P47 knockout mice transplanted with the wild-type bone marrow still lose just as much weight and still have just as bad histology scores in the context of DSS colitis. So we next interrogated what was the role of the microbiota in this context. And to do this, we simply heterozygously bred our mice. In doing so, we're able to compare P47 knockout mice with littermate controls. And in this process, from birth, both groups of mice have a homogenized or a standardized microbiota. And here we see that in doing this, we no longer see a difference in susceptibility to DSS colitis, either through the measure of body weight or by the evaluation of histology scores. Now, when we looked at GP91 knockout mice, well, it was a little peculiar. So for one, we looked at two models. We looked at the Citrobacter rodentium infection model, and here found that there was no increased susceptibility to colitis in GP91 knockout mice, neither as measured by body weight, histology score, or by differences in residual Citrobacter-densim CFUs in the stool. Similarly, when we looked at the DSS colitis model in this context, again, we did not see any increased susceptibility to DSS colitis in GP91 knockout mice. We did see an increase in the histology score, however, but when we took a closer look, we saw that this was largely related to increased proliferation of intestinal epithelial cells, likely related to an improved restoration of the intestinal barrier in the context of DSS colitis. When I moved to Montreal, we decided to reevaluate colitis susceptibility in GP91 knockout mice. And we did this using the TNBS model. And in here, what we see is, again, the microbiota really seems to play an important role. So what I'm showing you in this first experiment are GP91 mice that were heterozygously bred. And what we did is, at weaning, we separated the GP91 knockout mice from the littermate control mice, allowing for each of these mouse strains to sort of develop their own microbiota. And when we do this, we find that the wild type mice, as expected, recover from the TNBS colitis. Just as a note, the sudden drop in weight at day 7 is really related to the overnight fast prior to the intrarectal injection of TNBS. So wild type recover, and GP91 mice who are injected with a sham injection, they recover as well, of course, because they don't receive TNBS. But the GP91 knockout mice truly lose and continue to lose weight. Similarly, those mice have increased mortality and worse disease activity indices, which are a measure of weight loss, stool consistency, and rectal bleeding. In this second experiment, we did not separate our GP91 knockout mice from the littermate controls at weaning. So we left them co-housed, thereby continuing to homogenize the microbiota throughout the TNBS experiment. And here what we see is that the GP91 knockout mice that were kept co-housed with wild-type mice actually recover from their weight loss in the setting of TNBS. Whereas the GP91 knockout mice that were not kept co-housed with wild type continued to lose weight. And the wild type mice that were co-housed with the GP91 knockout mice, well, they actually continue to lose weight. So they look a little worse than they do than if they were separated at weaning. And this is further demonstrated here in the evaluation of disease activity index, where the GP91 knockout mice have a much less severe disease activity index when they're co-housed with the wild type mice. So we wanted to then take a closer look as to what exactly does the microbiota do to the intestinal epithelium insofar as ROS production. And here I'm going back to an experiment that was done at the NIH with the NIH mice in the context of the NIH microbiota. And as I showed you from my previous experiment in P47 knockout mice, you saw that the mice, P47 mice that were homozygously bred, with thereby maintaining their own genotype-specific microbiota, have increased susceptibility to colitis, and presumably they have a cholitogenic microbiota. And so what we see here is we took wild-type GP91 and P47 knockout mice, and we induced DSS colitis in them. We then isolated their intestinal epithelial cells and performed Diogenes assays, measuring superoxide formation in these sorted intestinal epithelial cells. And what we see is that there's an increase, a significant increase in ROS production in the P47 knockout mouse epithelial cells. Similarly, when we went and looked at NOX gene expression in these same intestinal epithelial cells, we find that the P47 epithelial cells have an increase in NOX1 gene expression, which likely explains this rise in ROS production. As a proof of principle to demonstrate that this was highly related to the microbiota, what we did is we took wild-type mice and then another group of wild-type mice, which at birth were cross-fostered with P47 knockout mice. And when you cross-foster mice, they acquire the microbiome of the fostering mother. And in this case, the wild-type mice truly were colonized soon after birth with the P47 knockout microbiota. And we've proven that this colonization was effective by sequencing analysis of the microbiome. And what we see is similar to the above experiment, the wild-type mice that are colonized with the P47 microbiome, well, their colonic epithelial cells also produce a lot more superoxide. And similarly, when we looked at gene expression, we see that those same colonocytes have a tremendous increase in NOX1 gene expression. So at this point, we decided that it was time to really have a look at the intestinal microbiota in our wild-type mice and our mice with CGD. And these are the colonies that were maintained at the NIH. And what we found, much to our surprise, were that GP91 knockout mice only were colonized or showed at least enrichment for bacteria in the Deferribacteries phylum. And when we took a closer look, we saw that, in fact, that entire phylum was accounted for by one bacterial species, which was mucisperamchidlari. So here you see that in the presence of in the fecal pellet in mice at baseline, mucisperamchidlari is present only in GP91 knockout mice. It is then slightly enriched in the fecal pellets of these mice after DSS colitis induction. And then we also looked at the microbiome at the level of the cecal mucosa, because this would be the microbiota that's the most closely interacting with the intestinal barrier. And what we see is an enrichment in mucisperamchidlari in GP91 mice at baseline, which is even more pronounced in the setting of DSS colitis. So we were intrigued by this, and we went back to some previous experiments that we've done where we've manipulated the microbiota in our mice to see if we could correlate some of the phenotypes that we observed with colonization with mucisperamchidlari. So here I'm showing you, again, this is all in DSS colitis models, a P47 mice versus P47 mice that were co-housed with GP91 knockout mice. This would mean that in adulthood, P47 mice would have been exposed to the GP91 microbiota and vice versa. And this is by coprophagy. So what we see in this case is that the P47 knockout mice that are co-housed with GP91 tend to maybe have a tendency towards losing a little bit less weight. They seem to have slightly less severe disease activity and a trend towards a less severe histology score. And surely enough, we found that those P47 mice that were co-housed somehow also got colonized with mucisperamchidlari. When we looked at GP91 mice versus GP91 mice co-housed with P47, we see that the GP91 mice that were co-housed with P47, who have presumably a more cholitogenic microbiota, tended to lose a little bit more weight, have a tendency towards not a real difference in disease activity index, not a major difference neither in histology score. And when we go and look at mucisperamchidlari colonization, we see, of course, that the co-housed GP91 mice, although they maintain some mucisperamchidlari, it seems to be a little bit decreased. So why is mucisperamchidlari so interesting? Well, it's a gram-negative, a strict anaerobe, a spirochete with a flagellum, and it lives in the intestinal mucus layer. Interestingly, this is not just a bacteria that colonizes mice. We went back and looked at sequences, microbiome sequencing analyses done and from several databases and in other reports. And we find that in studies where the microbiome was evaluated at the level of the intestinal mucosa, so not expressed stool, we find that this bacteria is actually present in 42% of those humans. Another point that's interesting is that mucisperamchidlari can degrade oxygen and reactive oxygen species. In the context of other studies, what we found was also quite interesting. So in the context of NOD2 and CYBB or GP91 double knockout mice, these mice, when colonized with mucisperamchidlari, develop a spontaneous colitis. However, in another study, colonization with mucisperamchidlari could actually protect against infection with Salmonella enterica, likely by competing for essential nutrients such as nitrate. So we went back and looked at our mice, and what we did here is we compared wild-type mice with GP91 mice that were colonized with mucisperamchidlari or not colonized with mucisperamchidlari. And this was clearly proven here by sequencing of the stool microbiome in our mice. And what we find, again, in the DSS colitis model is that mucisperamchidlari colonized GP91 knockout mice lose the least amount of weight, even a little bit less than wild-type mice in the setting of DSS colitis, whereas the GP91 mice that are not colonized with mucisperamchidlari lose the most weight. We also found that GP91 mice not colonized with mucisperamchidlari had increased mortality, which was not the case with those that were colonized with mucisperalium. And we find that the GP91 mice colonized with mucisperamchidlari had worse histology scores compared to those who were colonized with this bacteria. So at this point, we had to go and take a closer look at our mucisperamchidlari strain. So we isolated the bacteria from our mice, and we performed whole genome sequencing. And we compared the sequences from all the isolates from our mouse colonies in comparison to all the published isolates and the type strain of this bacteria, which we also obtained. And what we saw was that our, what we'll now call host-adapted Mchidlari strain had almost 10,000 different SNPs compared to the type strains. More specifically, we found that there were 321 unique genes expressed in our host-adapted Mchidlari strain. We then confirmed the presence of some of these unique genes by PCR. At this point, knowing that we had a different strain of mucisperamchidlari, not a different species, but a different strain, we then proceeded to do some biochemical or metabolic analyses to better understand if functionally our strain was different. So the first experiment we did is we co-cultured each of our mucisperamchidlari strains with exogenous hydrogen peroxide to see if there was any difference in the metabolism of hydrogen peroxide. And what we found was that our host-adapted Mchidlari strain metabolized hydrogen peroxide a lot more slowly than the type strain. We then looked at metabolic profiles between our two strains, and we did this using a biolog panel, which is a plate of several different metabolic substrates. And then we observed the metabolism of each of these substrates. And what we found was clearly that our host-adapted Mchidlari strain had a distinct metabolic profile, which seemed to favor increases in the glycosylate and decarboxylate pathways, the pyruvate and methane metabolism pathways. So our next objective was to determine really what is the impact of our host-adapted Mchidlari strain on the host. So to do this, we developed a co-culture system where we co-cultured intestinal epithelial cell lines. So we have mouse and human cell lines that we co-cultured with either Mchidlari strain. And then we isolated the epithelial cells and performed gene expression analyses. So what we found is that the incubation with the host-adapted mChedlari strain induced increased expression of CXCL2, the neutrophil chemoattractant, but it also, in comparison to the type strain of mChedlari, really did not induce very much expression of defensin alpha-1, an antimicrobial peptide, which in comparison is significantly increased in the type strain of mChedlari. Similarly, we found that there was a significant increase in Gasdermin 4 gene expression in our cells that were incubated with the type strain of mChedlari, but that was not the case in our host-adapted strain, suggesting that the host-adapted mChedlari strain perhaps does not trigger or promote as much NLRP3 inflammasome activation. Now, we know that it is often components of the bacterial cell wall that can induce inflammatory reactions in the host. One suspect would be looking at lipid A. And so what we did is, by mass spectrometry, we looked at lipid A profiles in both of our strains. And surely enough, we found that our mChedlari host-adapted strain had a distinct lipid A mass spec profile compared to the type strain. And now we're doing further studies to look at the implications of these different lipid A profiles in our host cells. To take this just one step further, and this is preliminary data, but thanks to a generous donation from Fabien Touzeau, we were able to do co-culture experiments between our mChedlari strain and his THP1 CRISPR-modified cells. So THP1 are human monocyte cells, and Fabien's group developed a cell line that had a knockout for CYBB. So we compared wild-type THP1 with CYBB knockout THP1 cells, co-cultured with each of our mucisperamchidlari strains. And interestingly, we found that only in the THP1 CYBB knockout cells that were co-cultured with our host-adapted mChedlari did we see that the host-adapted mChedlari really grew a lot more, or proliferated a lot more in this co-culture context. So this suggests that there's something about the milieu generated by the CYBB or GP91 knockout context that favors the growth of our host-adapted strain of mucisperamchidlari. So with this, we're sort of entertaining the following hypothesis. We think that for different reasons in early life, rather related to mode of delivery, nutrition, exposure to antibiotics, and previous infections, there are patients with CGD who manage to have perhaps an in-host adaptation of their microbiota, in which case the microbiota adapts to the NOX2 deficient environment and acquires certain properties that allow it to work towards maintaining intestinal homeostasis in this context. In comparison, there are other patients who perhaps, because of other intrinsic factors that I previously mentioned, might not manage to have this in-host bacterial adaptation. Instead, they have intestinal dysbiosis, which then engages a vicious cycle of intestinal inflammation. This is our hypothesis, and it's something that we're actively investigating at the moment. So now for the next part of my talk, we'll proceed to studies of the microbiota in patients with CGD. So our first question was to determine what is the impact of a NOX2 defect on the intestinal microbiota in patients with CGD. To do this, we wanted to really stratify our patient cohorts to have the least amount of confounding variables possible. So we know from interim analyses of the microbiota in patients with CGD that the biggest, by far, modifiers of the microbiota in this context are antibiotics. So not the prophylactic antibiotics, but actually what we refer to as the acute antibiotics. So any antibiotics used to treat an acute infection in the context of CGD. We also found that immune modulators, especially steroids, have a big impact on the intestinal microbiota. So in this context, what we did is we took samples from patients with CGD who were only on antimicrobial prophylaxis, so trimethoprim, sulfamethoxazole, and an azole, and were not on any other medication and did not have inflammatory bowel disease. And we compared the microbiota from the stool of these patients with that of patients that are healthy and patients who were on prophylactic antibiotics, more specifically trimethoprim, sulfamethoxazole, for prophylaxis against urinary tract infections or recurrent urinary tract infections. And firstly, we looked at the alpha diversity, so that's the richness of the microbial community. And of course, the patients that are healthy have the most microbial richness, which is associated with, better outcomes, less inflammation, more intestinal health. And similarly, patients who were healthy but on prophylactic antibiotics had very similar levels of microbial richness. However, patients with CGD had a decreased alpha diversity or microbial richness. We then looked at the beta diversity of our population. So that's the overall appreciation of the composition of the intestinal microbiota. This is demonstrated here by principal component analysis, where each circle represents a stool sample from a different patient. And what we see is firstly that samples from patients on microbial prophylaxis who are healthy cluster with samples from healthy individuals. But samples from patients with CGD cluster together and separately from healthy individuals and individuals who are healthy and on prophylaxis. We then looked more specifically at which bacterial taxa were significantly different when comparing patients with CGD versus healthy individuals. And in this case, positive values mean that they're more abundant in patients with CGD compared to healthy individuals. What we see is that there is enrichment for Blautia cassimiris and Anaerostipes cacae in samples from patients with CGD. But more importantly, rather than enrichment for species, we see that there is a lack of certain microbial taxa that in patients with CGD compared to healthy individuals. And some of the most notable ones are the Ruminocaucaceae, Lachnospira, and various species of Bacteroides. Now, we know that the intestinal microbiome produces metabolites. And these metabolites give you some indication of the function of the intestinal microbiome, and they can have an effect locally at the intestinal barrier, or more systemically by translocating from the intestinal barrier into systemic circulation. So here we looked at the metabolomic profiles from the same stool samples that we evaluated by sequencing. And again, we find that the metabolomic profiles can also distinguish healthy individuals from patients with CGD, as demonstrated in this principal component analysis. Looking more specifically in the volcano plot, we find that samples from patients with CGD seem to be enriched in the following metabolites, specifically carbamyl phosphate, L-threonine, fumaric acid, taurine, nicotinic acid, L-aspartic acid, and succinic acid. When we look at these differentially represented metabolites, we see that samples from patients with CGD seem to have this predisposition towards alanine, aspartate, and glutamate metabolism, as well as for nicotinate and nicotinamide metabolism. And the implications of this physiologically are currently being evaluated. So our next question was whether or not the intestinal microbiome could distinguish samples from patients with CGD IBD. So here we stratified our samples for acute antibiotic use. And what I'm showing you are the samples from patients who are only on antimicrobial prophylaxis, no acute antibiotics. And I'm comparing samples from patients who have never had CGD IBD with samples from patients with active CGD IBD. And when we look at the alpha diversity or microbial richness, we see that samples from patients with active CGD-IBD are less rich. There's less microbial diversity. When we look at the beta diversity or the microbial composition, we see that samples from patients with active CGD-IBD clustered together and separately from samples from patients who do not have CGD-IBD. Again, when we looked at the specific bacterial taxa that were differentially represented between these two groups, we see that patients with CGD-IBD are enriched for Veillonella, for Streptococcus mutans, for Proteus, and for Enterococcus, which is not unlike what we see in the general IBD population. We also see that they are somewhat poor in certain bacterial taxa, like Prevotella copri, like Phascolarctobacterium faecium, Megasphaerum massiliensis, as well as Coprococcus comes, Bacteroides cacae, and Alstipes. So our next objective was to look at the impact of certain treatment interventions on the intestinal microbiota in patients with CGD. So one of these interventions, now this was done in one patient, so it's a case report, but it's very interesting. We had a pediatric patient with CGD-IBD who we treated with an elemental diet or exclusive enteral nutrition. And what we found is that not only did this induce sustained changes in the intestinal microbiota, but it also induced clinical remission, which was then sustained with azathioprine long-term. And this patient never required steroids again. And we followed him now for at least 5 years. And that's impressive, considering that this patient was on antimicrobial prophylaxis and at certain points likely received some acute antibiotics for specific infections. So specifically focusing on the microbiota, here I'm showing you again the alpha diversity, and what we see here is there is modest microbial richness pre-elemental diet and during the elemental diet. However, the microbial richness and diversity increases after the elemental diet, rendering it comparable, in fact, to the richness of patients with CGD who have never had IBD. Of course, it's still not quite as rich as that of healthy individuals, but nevertheless, it has improved. In the next part of the figure, I'm showing you the microbial composition or the beta diversity of these samples. So here in red is the patient sample pre-elemental diet, and we see that the microbial community structure shifts during the elemental diet, and then it further shifts again after the elemental diet. And this is what's interesting. These are all samples collected over the course of 5 years, and what we could see is that these samples cluster together. So they are the most similar. In comparison, I'm showing you in yellow samples from healthy individuals and samples from patients, that's in yellow, and samples from patients without IBD in pink, and samples from patients with CGD-IBD. So the samples from the patient post-elemental diet most resemble those from healthy individuals and individuals with CGD without IBD, but are significantly different from samples from patients with CGD-IBD. We then had a look at the exact microbial taxa that were differentially represented between the samples from the patient post-elemental diet and in comparison with samples from patients with CGD-IBD. And now we see that with the elemental diet, the patient has enrichment for Faecalibacterium prausnitzii, Dialister propioni faciens, and Parabacteroides merdi and Blautia, to name a few, all of which are bacterial taxa that have been associated with intestinal health. We also see in comparison decrease in bacterial taxa that are not typically associated with intestinal health, such as Escherichia and Shigella. So We know that CGD-IBD resolves after hematopoietic cell transplantation. And this was beautifully shown in a paper from Rebecca Marsh and all members from the PIDTC group, so the Primary Immune Deficiency Treatment Consortium, specifically from Protocol 6903. And within this context, we wanted to look to see if allogeneic hematopoietic cell transplantation in patients with CGD could actually shift the intestinal microbial community structure towards one that resembles that of healthy individuals. So here I'm showing you the alpha diversity or the microbial richness and diversity of our samples. And I apologize, it's not necessarily in the chronological order of the transplant, but nevertheless, the findings are quite interesting. So here are the samples from patients at baseline pre-transplant. These are samples from healthy individuals. So clearly, there is increased microbial diversity and richness in samples from healthy individuals. The samples from patients at baseline resemble those of patients on day 0 of transplant, so day of transplantation, that is after conditioning. And the diversity remains pretty constantly moderate to not great in the early phase of the transplant, so day 7, day 30. However, as of 6 months, we see a significant increase in the microbial diversity and richness, which is then sustained at 1 year and even improved 2 years post-transplant. Here I'm showing you the beta diversity or the microbial community structure. So these are samples from patients at baseline in red, and they are clustering with samples from the early transplant phase. So that's in purple here. So that's day 0, day 30. And also day 0 in orange, sorry, day 7 and day 30 are the purples. And then you see that as we get into the late transplant phases, that is 6 months, 1 year, and 2 years out, that's these green samples, there clearly begins to be a shift in the microbial composition, and it tends to migrate towards the composition of samples from healthy individuals. When we look specifically at the microbial taxa that are differentially represented between patients 1 to 2 years post-transplant versus patients with CGD at baseline, we now see that post-transplant, there's enrichment for some very key bacterial taxa like Ruminococcus callidus, Veillonella parvula, Coprococcus comis, Alistypes, Doria. And these taxa that I'm pointing out with the red arrows are those which we know from our previous work that were really poor and underrepresented in CGD samples at baseline, and even more underrepresented in patients with CGD and IBD. So clearly the transplant altered the microbial composition and enriched taxa that have been typically associated with intestinal health. So here I'm showing you the taxa that are differentially represented between samples from patients 1 to 2 years post-transplant and healthy individuals. And here what we see is that, of course, the microbiota in CGD patients post-transplant is not exactly like that of healthy individuals. There are still several taxa that are differentially represented. Nevertheless, there is certainly a shift in the microbial community structure. And I'm pointing out here that there is still a bacterial taxa that remains underrepresented even post-transplant, which is the Bacteroides nordiae. So in conclusion, the microbiome established at birth significantly impacts colitis susceptibility in mice with CGD. We also know that the NOX2 deficient environment may lead to in-host adaptation of bacterial strains, which may then acquire new functional and/or protective properties. Patients with CGD have distinct intestinal microbiome signatures, which further distinguish patients with CGD-IBD. Treatment with an elemental diet may help induce clinical remission from CGD-IBD in association with sustained changes to the intestinal microbiome. The HCT, intestinal microbiome signature, shifts towards that of healthy individuals with associated increases in protective bacteria and decreases in chalitogenic bacteria. And with that, I'd like to acknowledge several people who have really made this research possible, members of my laboratory, people that I work with at the NIH still, members of the Holland laboratory, additional members from NIAID, members from NHGRI, especially Julie Segre, lots of collaborators across Canada and the US, and of course, the members of the PIDTC CGD working group and steering committee, and all the funding agencies that have supported our research. So with that, thank you, and thank you for your attention.